And welcome everybody to this webinar. My name is Brenda Andrews. I'm currently Vice President of the Genetic Society of America, and I'm a professor at the University of Toronto. And I'm here today to introduce Paul Sternberg, who will be giving a lecture in our 2024 GSA Awards seminar series. And so as in March of this year, Paul was presented with the Thomas Hunt Morgan Medal at the Allied Genetics Conference 2024. And this award honors a GSA member's lifetime achievement in the field of genetics, recognizing their full body of work. So I'll just say a few words about Paul's career and his work in C. elegans genetics. He was recognized with this medal for his far-reaching contributions to the field of genetics, developmental biology, evolution, neuroscience, and disease through the use of C. elegans as an experimental system. His interest in C. elegans as a model system began during his PhD thesis work with Robert Hurwitz at MIT and continued throughout his postdoctoral studies, even though he made a productive foray into yeast genetics while working with Ira Herskowitz at UCSF. I also studied with Ira and was lucky enough to overlap with Paul and land in the lab while he was still a member and had just published some beautiful work identifying suppressors of SWI or SYN mutants in yeast, which turned out to be a treasure trove of genetic discoveries. And it really set the stage for my own career in yeast genetics, as well as many others that were in the lab at the time and after us. Paul, however, returned to his roots in C. elegans development when he started his lab at Caltech, where he's now the Thomas Hunt Morgan Professor of Biology and an investigator of the HHMI. He really is continuing to try to understand how the genome controls development, behavior, and physiology, or as his title of his talk indicates, basically continuing to explore the genotype to phenotype relationship. And he has really made abundant use of all of the accessibility of C. elegans to pursue these interesting questions. And he's also been working on parasitic and pestilent nematodes as well. And Paul has really been a central member of the worm community over the years, and particularly to the development of tools like Wormbase, the Alliance of Genome Resources, and the Genotype-phenotype Consortium, where he is a founding member of Micropublication Biology. The Wormbase, of course, is very important. It's an international consortium of biologists and computer scientists who make available information on the genomics, genetics, and biology of C. elegans and related nematodes to the research community. These resources, I'm sure, are used, as well as the Genotype-phenotype Consortium resources, are used every day by many of you on this call and scientists throughout the research genetics community. His lab also continues to organize, store, and display information about C. elegans and extend these efforts to other nematodes. This is done on Wormbase, and he's also developed data sharing processes like Expresso, which is a search engine for the biological literature, and has applied it to the literature of C. elegans, Drosophila, Arabidopsis, and others. So these are really resources that take a lot of work to maintain, and for the sole purpose of making sure that everybody in the genetics community can have access to information as soon as it's produced. And if you have questions for Paul, please put them in the chat and we'll get to as many as we can at the end of this presentation. If you want to ask a question anonymously, you can direct it to me or Jessica in the chat. Paul, look forward to your talk.
Thanks, Brenda. It's good to see you again. And and see everybody here who I don't see their screen. While you were talking, there were 302 participants. It's gone up since then, but that's exactly the number of C. elegans neurons. I'm really grateful to the Genetic Society and the nominators and board and all the members, and for this really lovely and great honor to me. So I want to talk about some aspects of a fundamental problem in development, which is how do you understand the properties of an organism from its genome. And you wouldn't have known that, but actually that was C. elegans genome sequence. And if we could read it appropriately, we would know that it was C. elegans. Now, some of the work I'm going to describe has been done recently and some over the years to try to highlight some classic experiments by a great set of students and postdocs and research assistants, and as well as high school students and undergraduates and visitors. So I'm really grateful to them, and I'll mention some of the contributions as I go through. Okay, So the problem really is, can we predict the phenotype or attributes of the organism genome sequence? Now, there's a lot of aspects to that, not that I won't all cover today. One of them is, what's the effect of a variant, right? This is very important for human genetics and, even for experimental experiments in the lab where we induce mutations and we want to understand the variant. There we're going from phenotype to variant. We're also obviously going from variant, especially in human health and disease, to phenotype. Another one is to predict cell properties from, say, a transcriptome or proteome. So how do you really think about a set of 5,000 or 6,000 genes or proteins in a cell? Do we know what the cell is? A practical consequence of that would be, how do you understand the meaning of a set of differentially expressed proteins? And I'll try to give you my perspective and some examples and, where things are, which is we have a long way to go. Okay, so what's the consequence of a single nucleotide change? Okay, here's a single, G to A change in a gene in C. elegans, and it was induced by EMS. And the properties of this was that it actually blocks two aspects very specifically of the male copulatory behavior, where the males first recognize their mate, and in the absence of, in this mutant, they fail to contact the hermaphrodite and just pass on by. But it's incomplete penetrant. So if they do respond, then they continue backing along the hermaphrodite, turning beautifully, and then they're unable to officially find the vulva. So this was Maureen Barr's work, where she figured out some of the genes, the polycystin 1 and 2, or PKD1 and 2 genes in human were involved in this. And this was Alison Whitaker had found by a phenotypic screen another allele of this gene, which Maureen had knocked out in a reverse genetic approach. So that's kind of one, any one variant can be really important or not. There's a lot of work from many of this on the call and around the world about trying to understand in model organisms the effect and importance of individual variants. It's definitely a growth area. Okay, what about the genome? Well, here's a really cool behavior. Here's insect-killing nematode, Sternberg carpocapsae, and it can jump. So if you put on host odors or CO2, it's gone in one frame. Let's slow that down. It's going to jump up here. This is a paper published in Micropublication Biology. And that worm can get to the lid of a Petri plate and back. Okay, here's a video rate again. It's going to jump down. There it goes. One frame later, it's gone. Okay, so we had this Adler Dillman who came to the lab as a graduate student, said, I want to understand this. We thought, well, we'll get a genome sequence and that'll help. So he, along with Ali Mortazavi, sequenced genomes, draft genomes, early on, 10 years ago, of 5 different Sternberg species, carpocapsae shown down here. And only in this part of the clade are there these jumping ambushers that can jump. So the fantasy was you could understand that from the genome sequence. Well, here's, to make it even harder, here's C. elegans versus carpocapsae, looking at how many protein domains of various types are present in the genome. And this is a good idea. This is like looking at 2 different languages and trying to understand what that culture thinks is important. So C. elegans has lots of these serpentine receptors, G protein coupled receptors that are olfactory receptors, many more than the carpocapsae, or different types. There's a huge difference in protein domains associated with regulation of protein proteolysis. And one thing that's really expanded in carpocapsae, which was kind of expected, were proteases and protease inhibitors. So now if we kind of look at a comparison in just this clade, this genus, you can see there's a reasonable correlation of the number of this particular trypsin family serine proteases and the reported host range. So Skaptarisky, which is supposedly a cricket specialist, has one of them. Carpocapsae, which is more generalist, has 9 genes. Okay, that's a hypothesis, a lot of hypotheses. But in terms of understanding jumping, it didn't really help yet, but it does provide a lot of tools. But I think it just kind of gives you another example of, why we have to understand genotype to phenotype. Okay. Well, here in 2024, a lot of people think that AI can or will or is solving all our problems. So, what's my take on that? Well, clearly for protein structure, AlphaFold's been stunning in terms of being able to predict many proteins, their structure, and that's, it's really, world changing. Now, it's generally recognized that the reason that AlphaFold, large computational model, was successful because of the curation in the protein data bank, which is a worldwide effort, PDB. So that curated information of high quality information of many structures, tens of thousands of structures, allowed the computation to finally break through and give good answers. Okay, so we're going to assume that AI works. Now, will it work for the genotype to phenotype problem? Well, we don't really know yet. But for example, if we put all the information, curate all the information that exists in the Alliance of Genome Resources, worm-based, fly-based, yeast database, et cetera, could we come up with a program, G2P, that would do the equivalent? Okay, Alliance is really very useful now because alliancegenome.org is giving you one-stop shopping for a lot of the information that's been in the major model organism databases, as I mentioned. And this includes lots of information: genome sequence, variants. We're getting in sequence features, genes, the gene ontology annotations, some beginning of pathway information, phenotypes, connection to disease, anatomy, et cetera. So that's a lot of information. But there's a nice study that's in the works from Paul Thomas and the Genontology Consortium, Paul Thomas's group, shown here, was taking all the annotations from gene ontology annotations from all organisms. So they're basing it on, I think the consortium is basing on almost 200,000 papers and all these specific annotations. They're looking at phylogeny, how it compares, and they did a very nice analysis of the different functions. And the bottom line of this paper, which is in the works, is that 80% of human genes can be assigned at least some functional characteristic, like where it is in the cell, what's its molecular function, or what kind of process it's involved in. About a third of the genes, they're calling functionally well characterized, meaning it has at least one of each. It has one of each of those categories. So you could say some story about that gene. Now, I would say that for a lot of those genes, it's not a complete enough story. So we have a long way to go. But as a sideline of this analysis, we found that 2/3 of the information about human genes comes from model organisms. So this is, I think, a strength of the Genetic Society of America and all of us, which is using model organisms to understand human genes. But anyway, we don't have enough information. So I would say if we want to really compute through a curated resource to genotype to phenotype, we need more data. And that's what I'm going to, focus on today. Like, what are those kinds of data? How are we doing? Okay. Right. And for most genes, if you go to the Alliance, you look, most genes will say things like molecular interactions, no data available, genetic interactions, no data available. The descriptions are pretty much, it's a G protein coupled receptor. Okay. So that's useful, gives a hint, but we need a lot more information. Okay. So, there's a lot of, okay. Guess I missed a slide here. Right. So the basic kind of information you want is, you want to know what's the phenotype of a loss of function mutation. And for C. elegans, there's 20,000 genes, and we have knockout strains for about 8,000 of them. So it's pretty good. About 1,000 of those are from, in the recent few years, from Anne Rugby at the Sinoraidatis Genetics Center on the Knockout Consortium, of which we've been helping a little. And I just want to say, the approach we were taking, which was developed by Han Wang and Jonathan Liu and Hee-Nam Park in a G3 paper in 2018, is to do the knockouts by inserting a universal knockout cassette. So the idea is we cut and then insert 43 nucleotides using homology-directed repair, and that cassette has stop frames in all stop codons in all frames plus frame shifts 2/3 of the time, and that will induce an early stop. And it also has an exogenous efficient Cas9 target site with a PAM site, so you can do re-editing. So you don't have to even think about what reading frame it is. Just stick it as early as possible in the coding region where there's a good PAM site, and you can analyze it by PCR or RFLP mapping. And the other advantage is you can re-edit it, because if you have this mutant strain and you want to make an isogenic control, the best thing to do is cut again and repair back to wild type, so you have a true revertant. And then you can compare the strains, and the only differences are the, one mutation per generation that you get from spontaneous variation. And it's a reproducible lesion. So if you're going to construct a double mutant strain, you can do it independently and you're going to get the same event. So that's been very useful. And I'll just show how we've applied it to one nice biological problem, which is the diapause reproductive decision in C. elegans. So as you may know, C. elegans, when they hatch, these baby L1 larvae, and during the L1 and L2 stage, they decide whether to go straight to reproduction in 2 days, or if conditions are unfavorable, they go into this Dauer larval state, which can persist for months, and then they can exit rapidly. The induction of this depends on how much food there is. So low food, high crowding, high temperature, other stresses induce Dauer formation. When we did an RNA-seq analysis years ago from Oren et al., published in this PNAS paper 2018, we found that strikingly, all the neuropeptides were upregulated during the progression into Dauer. So using a modification published assay from the 1980s, we titrated the pheromone concentration to a point where you would get, which is the crowding signal, where you get about 50% Dauer. So in each experiment, you're trying to get about 50% Dauers, and then you ask, does that genotype matter? And so even a twofold effect would show up very nicely. And from that analysis, we found that there was a handful of these neuropeptide genes that promoted Dauers and others inhibited Dauer formation. So Cynthia Tsai came to the lab and, as part of her project, decided to really do a systematic analysis at the receptor level. And using the stop-in method, we made another 36 mutants in putative neuropeptide receptors. And there's a total of about 93 out of the 100 or so receptors that she assayed, and using this sensitive assay. And about 30 to 40% of them actually had phenotypes, and they were either increased or decreased diapause. So, that's, kind of stunning, and it shows the sensitivity of the assay. But then combining this with gene expression data from Seth Taylor and David Miller and Oliver Hobert and Mark Hammerlin's groups, she was able to do a general analysis of how the brain computes information about the environment for this very important life cycle decision, which is kind of a neuroeconomic decision about whether they should reproduce or wait to reproduce. And so if we have neurons on the left and these receptors with phenotypes at the top, you can split them into pro-growth or pro-Dauer. And it's clear it's sparse. And any given neuron has typically multiple receptors. And when they have multiple receptors, as shown here on the right, those receptors have opposite signs of action. And each receptor is expressed in a handful of cells. So it's very complicated, but it suggests that the whole brain is feeding into this Dauer decision. So information from the entire body over this 12 or 14 hours is being used for this decision. Now, these knockouts then, of course, allow lots of specific experiments that'll take, years, decades to sort through. Okay, back to basic information. Well, we talked about phenotypes, and gene ontology terms. Now I want to talk about interactions and sort of how you develop pathways. Okay, so now I'm going to go back to some classic work, which is where I started my lab and worked on as a graduate student with Bob Horvitz, was C. elegans vulva development. And this diagram taken from Worm Book, which was we started and then just was nicely taken over by the Journal of Genetics at GSA and is continuing now, is there's precursor cells that can give rise to the wild type vulva, which is involved in egg laying and copulation with males. And it depends on signal from the somatic gonad, from the anchor cell. And then there are these, in this case, 3 different cell types of precursors. And this is developmental patterning, which I could go on about for hours, and some of you in the room have heard me do so. But if you remove that anchor cell, There's no vulva. The cells are just making epidermis. And Sulston and Horvitz, in groundbreaking work, showed that there were two classes of mutants you could find. One was vulvaless, which had all the cells, but no vulva formed, or multivulva, which even, as Chip Ferguson showed, that even in the absence of the signal, that is, it's laser ablated, that you could get extra vulval tissue. So using those genetics, I'll just give an example of ligand receptor. You can do, from 1990s, we did these epistasis experiments. So the top is the pathway. Lin3, EGF, goes through Let23, EGF receptor to give a normal vulva. Knock out Lin3, vulvaless. Knock out Let23, vulvaless. Activate overexpressed Lin3, the ligand, multivulva. Activating mutation of the receptor, multivulva. Activate the receptor, knock out Lin3, still multivulva, a little less so. Increase Lin3, knock out Let23, it's vulvaless. So that all kind of gives you this, opposite phenotypes. You can order the genes in a pathway. Right? And you could do that sequentially. And we spent a lot of time doing that, and it was fun. Okay, another problem. 10 years later, 2000s, there's another vulval problem, which is there are these secondary vulval precursor cells. We go back here, shown that actually have, when they divide and generate progeny, they have opposite polarity pointing towards the anchor cell. And this is nicely shown from some reagents that Takao Inoue and David Sherwood made, which is showing the inner and outer vulval cells. So there's this polarity. Okay, and there are these mutants, again, going back to early screens from Ferguson and Horvitz, in genes like Lin17 and 18, which gave this reversed posterior precursor cell. So you have a normal vulva from two of the cells and a reversal, so you get an extra little invagination. It's going to be a bump. It was called bivulva at the time, or reverse polarity. Okay. Now these mutants were incompletely penetrant. So in systems biology terminology, we can do the analysis like this. So this is going to be proportion wild type. Wild type animal, 100% normal. Mutant and ligand, one ligand, so Wnt protein, normal. Other ligand, almost normal. Receptors have incompletely penetrant phenotype, 25% normal, 60% normal. So you make all the 6 double mutant combinations, and what's shown in green, sorry, I should switch it to yellow and purple, but receptor 1, ligand 2, you get pretty much, statistically indistinguishable from the single mutants. So they kind of buffer each other. So they probably work together. Same thing with ligand 2 and receptor 2, you get the same phenotype. If you combine ligand 2, ligand 1, ligand 2, receptor 1, receptor 2, ligand 1, receptor 2, receptor 1, you get a more severe phenotype, and summarized here. So this says ligand 1 and receptor 1 work together, ligand 2 and receptor 2 work together, and they converge on a common output. So we could say this: one Wnt works through this frizzle type Wnt receptor Lin17, the other one goes through Lin18, which we argued at that time was a new type of Wnt receptor, which has held up pretty nicely. So that's kind of how do you get some critical pathway information from genetic interactions or epistasis. Okay, so back to basic information. We've talked about genes. We have about 8,000 null genes. Phenotypes, I'd say there's so many phenotypes that we only really have a very sparse information about phenotype. Gene expression is from single cell gene expression studies is getting quite good now. There's about maybe a third of the C. elegans genes have some reporter gene assays described in the literature from worm-base. For gene expression, most genes have RNA-seq and a lot of single cell data, and that's still not covering all stages, but it's gives some good hints. But again, cells will express 6,000 or 7,000 genes, so that's we need a lot more information. I just talked about interactions, and I would say that actually, the cases that are intensively studied, we've generated sort of scaffold and quite useful genetic pathways that can get fleshed out into really kind of more mechanistic pathways, but that's only in a handful of cases. And I want to mention the site of action, which is where does the gene act? and there's a difference in where it's expressed from where it acts. And I'll give an example of that from our recent work. Okay, so James Tan with Tingyi Wang here in our proteomics facility did this kind of proof of principle experiment. The question was, could you take small amounts of material and get proteins out? So James dissected out intestines from a single C. elegans young adult. Here's an intestine, and then process it for mass spectrometry, proteomics, and the same thing with a gonad or half a gonad. And you can get from a single gonad or intestine, you can get 3,500 proteins, which is quite good. And if you combine them and combine the individuals, you can get to the 4,000 or 5,000 proteins. So now analysis that there's lots of great information. One of the striking things was that there's about 100 proteins that were present in the gonad. But if you look at the RNA where that's made from various publications, some very nice RNA-seq data sets, they seem to be only made in the intestine. So there are proteins that are made in the intestine transferred to the gonad. Now, 6 of them we already knew about. They're the yolk proteins of vitellogenins, and they're the most abundant proteins in both tissues, as you'd expect. But then there's another 90 or so that are there, and the function of those are to be explored. So it's really important to, as many of us know, understand the difference between where gene acts and where it's expressed. Okay. So, it's, if you think about yeast for, I don't know, 15 or so years, some of the work of Brenda Andrews and the Toronto crew, you can get plates with knockouts for all the genes in yeast. In worms and flies, that's not available. Worms are, pretty nice. You can freeze them and in small vials and keep them for 20 years or so. But we don't have a really good way to have a whole collection of worm mutants and screen them in a sort of high throughput manner. There's some progress on microtiter culture and conditions and screens, especially with RNAi, but they're it's still a specialty. So I think that the random screens are still important to try to find interactions. So I want to give a recent example of a new method that Hillel Schwartz has developed that I think is very promising, and it kind of hints to the fact that there's lots more information that we could get. So his idea was to essentially do what's used in human genetics. You take phenotypic animals, organisms, and you look at their genome. So the idea is you could take mutagenized C. elegans, look for phenotypes of interest, take that individual, sequence its genome, and then if you look at enough examples, you can figure out what are reasonable causal variants, and then you can test them, because now that we can do genome editing, you can edit that variation, the variant, back into the genome and look at its phenotype. And he wanted to look at what a screen that would have been essentially impossible. So we'll talk about modifiers of LET23 EGF signaling. Okay, the concept on the screen is, well, okay, classically, there are 2 types of screens done in C. elegans. One was the original Brenner screen, where you mutagenize, let the worm self-fertilize, look at F2s that are of interest, they're viable and fertile, and you can study them. If you want to look for lethal or sterile mutations, or also very low penetrance, you can do a clonal screen where you set up F1s or F2s that are heterozygous. For example, F1s that are heterozygous for mutations, and look at their F2s. You can find lethals that way, or if it's incomplete penetrant, you can pick F2s and look for F3s. Okay, and you can study them. The aclonal screen is, again, you can even a dead animal, put it in a tube, process it, and genotype it. Okay, so the biology here is, I told you that EGF receptor LET23 was required for vulva development. Actually, the loss of function mutations that Bob Herman published in the 70s in Genetics, and we obsessed over in the early 90s, is lethal. So Hillel wanted to look for suppression of lethality, but we knew that if you let23 stimulates different signaling pathways, and for example, if you activate RAS, which would then restore vulval formation, it'll make the animal alive, but they're sterile. So the idea was screen for animals that the homozygous let23s are dead, pick animals that are less dead, or they're adults and they're sterile, and then study them. Okay, so you have a balanced strain with a good, nice GFP balancer. You mutagenize, let them self-fertilize, collect eggs, let them fertilize, and then you look for animals that are homozygous for this let23 lethal with a roll6 marker, which makes the animal have a twisted cuticle and it rolls. So you look for non-GFPs that are rolling and then sequence them if they're not viable. Here's an example. Here's a dead animal, very characteristic stage, and here's a not quite viable strain, but an animal that lived long enough to grow up and roll nicely. Okay. Now this actually is the reconstructed animal. So this is in a gene tensin 1, and it's a particular variant, S42F. And this gene from the original screen was suppressing the lethality of let23. If it's added back to the genome, it's viable, and you can show that it still suppresses let23. So that's a good hit. Okay. Right. And another variant was, so that one was of interest because 3 of the worms that were less dead had a mutation in a very specific exon of this tensin 1B isoform. So that looked like a good recurrent mutation, because there's there's a background of hundreds of mutations per mutagenized worm. You have to find recurrent hits, just like in human genetics. Okay. So, but another one was a specific point mutation in RAS that is this R164H, worm number 27. And the existing activated RAS mutations were all mostly in codon 13 and one of them in codon 19. And those are known in human cancer. This other R164, this is human and C. elegans RAS, which are very highly conserved. And this is another conserved residue. And in the literature, it's been shown in some very small % of cancers to be present. So activated RAS is very important in promoting cancer progression. And this is a very minor one. We didn't get out of other screens, but we found it in this case. So we feel from the reconstructions we've done of a dozen or so genes, and particularly this one, we're getting the right type of genes. So we believe them. So what are they? Okay, so I'm going to show you this, finish up with this pathway diagram where the proteins present in black are the core pathway and regulators thereof. So Lin3 EGF receptor, GRAB2 SOS, which activates RAS. RAS goes from GDP to GTP, stimulates the MAP kinase cascade through RAF, Link45, the BRAF homolog, MAP kinase goes to the nucleus, phosphorylates transcription factors. There's accessory factors that help or that are implicated in it, like the Lin2710 complex, which is involved in localization of the receptor. Now, shown in red are hits from the screen that are verified. So RAS is one I just mentioned. This tensin 1B, which is has a signal transduction functions in other systems. These ROGAF and ROGAF interacting proteins, that kind of makes sense. If we look in the literature, then there's this GAI1 protein that looks like it interacts with tensin and inhibits Rho, so it's a ROGAP. So that should act antagonistically to this. So it kind of smells right. Very recently, like last week, Hillel did an experiment and showed that this actually does have a phenotype in this signaling pathway. So that's quite predictive. Transcription factors always show up and are reasonable. I think the most surprising thing from this was a mutation in HIM14 MSH, which is a DNA damage repair enzyme. So we think there's some connection of this to DNA damage that we can discuss another time. So this tells us a couple of things. One is, if we look more intensively and do screening more intensively, we'll find more proteins. And this kind of matches our expectation. If you do proteomic studies, for example, immunoprecipitation of a protein, we haven't done it with the EGF receptor, but with other proteins, you get, hundreds of proteins, and it's very hard to actually go and test all of them. When you do, you find, there's a reasonable amount of having functions, but you have to look pretty hard. So here, with a very sensitive assay, we're getting, a large set of interacting proteins, and it points out that we have lots more work to do to understand the cell biology here, and be able to really understand, for example, if you had a variant in one of these, what it's going to do. So we've kind of, maybe doubled the number of interactors in this pathway almost from this one screen. And I think it'll be interesting to see what'll happen if we do similar methods in other cases. Okay, so let me kind of summarize here what we got. I think, the genomes define the genomes and omics studies that defined the parts of a network. There's a lot of, high throughput studies and individual studies that kind of make connections among these parts. And, you get some structure out, and that's, still lots of work. We need more of that information, as I mentioned, in terms of predicting how things work, but it doesn't give us the abstract, mechanistic understanding we like to do, which would be realize that this makes up a worm. We need more information. So I think what we have to do going forward is, do more genetics. I spent 40 or so years doing genetics with the worm. There's lots more to do. It's getting exciting. The tools are better. And more is illuminated. There's lots of connections between disparate genetic studies that, bring out delightful insights. I think it's really important, if we want to use artificial intelligence, to get information, so data and metadata, into knowledge bases like the Alliance Genome Resources. And then the other thing is to apply our real intelligence to understand, to real intelligence about what is the organism trying to do. So if you go back to the Dauer example, where there's many, many receptors, it sort of says to us, we really need to think about what information is the organism getting, what is it computing to try to make this decision, because that's what the genetics is telling us. So the feel for the organism is really important. High throughput studies are important. Mechanistic, detailed obsession over experiments are important. And genetics, of course, is still an amazing thing to make discoveries. So I want to say I've really enjoyed doing genetics and worm genetics and some yeast genetics over the years, and I look forward to many more years of continuing to do that, and also watch and listen to everybody on this call and what you discover. So thank you for your attention.
We do have one question in the chat already that I'll start with. And then, as we mentioned, people on the call, if you have questions, you can just type them in the chat and I'll field them over to Paul. I don't know if you can see them, Paul, anyway, but Jeremy Thorner put in a question right after you presented your jumping worms. And he notes that jumping requires musculature. Were there no obvious differences in actin and or myosin gene number or characteristics between the two nematode species?
So no, so the other thing about, okay, so yeah, I think we didn't really see anything with muscle and actin, but I think it's going to be, okay, if you said, how would we engineer a non-jumping worm to jump? Okay, so actually C. elegans, I kind of gloss over this, but the worms are standing on their tail doing a behavior called nictation. And C. elegans actually does that. The Dauer larvae stand on their tail and they wave back and forth. And it's actually it's a dispersal mechanism. So Junho Lee did what I first thought was a silly experiment, but actually is like profound, where he had a mutant at Seoul National University. He had a mutant that didn't nictate, and he said, does that help in dispersal? So he takes two Petri plates, and he puts worms on one and nothing on the other, and he waits an hour, and he asks, do they get from one Petri plate to the other? And they don't. And then he adds fruit flies to the cage, and the worms can get from one Petri plate to the other.
Mhm.
And then he puts in the mutant that doesn't nictate, and they don't transfer, and they actually can see them on the flies. So I think they're already like interacting from that stage with the insect. Okay, so now how do they jump? Well, they're on their tail. To get the jump in that video, what they're doing is they're taking their head and pressing it against their body, sliding it down to the substratum, and they kink their cuticle. And the original paper published, I think, in the 80s in Nature that described the jumping, argued that it was that the kink in the cuticle was a way of storing the information, the energy, and you're going to get, like snapping shrimp, the energy from muscle contraction stored, so you can get much faster than muscle contraction response, because it's really one frame it's there, one frame it's gone. So I think we have to look at the nervous system and how the wiring is changing, so you get this very pronounced ventral cut, a ventral bend. The other thing is, actually, I think they have to have some way adhesion so that they can that the head can stick to the body, and we need to look for that. Anyway, long answer.
Really cool phenotype. You want to read them, Paul, if you can see them? Or?
Okay, I don't know if I should. Okay, so I'm a grad student at Rutgers with Monica Driscoll.
Actually, so the next one was from Mira Sundaram. Do you see that one? I can read it if you like.
Yeah, read. Okay.
Interested in new LET23 suppressors. And the first question is, what do they die of? The LET23 mutants. And then there's a second component of that.
Yeah, so Mira, I don't know, but I'd love to talk to you about that and figure out how we can sort out the, what's really going on, because I think there is that other focus from Krog et al.
Yeah. And it says, do the suppressors act by restoring RAS-ERK activation?
We haven't looked. We're just trying to, prove that the genes actually were suppressing. Those good questions.
Yeah. Maureen is next. Maureen Barr, cool proteomics on single intestine and gonad. Any thoughts on how intestine is transferring the proteins to the gonad?
So, yeah, so the yolk is one that's been studied, and vitellogenins, and presumably they're carrier proteins. And actually, I don't know the literature on that as well as I should. Someone else can type in the answer. But there should be, the real question is, for the other proteins, is it the same mechanism or not? And we haven't looked at all. The list is out there, and, welcome to see what you think.
Yeah, a technical question from Michael Elian. To sequence individual worms, what kind of amplification do you do? What kind of coverage do you get? Okay,
so the coverage was about 50%. So we had the starting mutations, the LET23 mutation, the ROL6 mutation in there, and about almost exactly 50% of the animals had those changes. So that's our coverage. We're sequencing to about 10-fold, 5 to 10-fold. With the 50% amplification means that about half the time we might be missing the variant unless there's more than one. So it's worked out a lot better than what we expected. And Hillel and I talked about that. We thought, well, if we get a couple of hits and one of them looks good, we'll be stunned. And actually, we have a dozen hits that look really good. Okay, the loss of the DNA repair. Okay, so our argument about mutagenesis is that, given that there aren't that many cell divisions from, in C. elegans, that the DNA repair, somatic mutations, we don't think is going to be the problem. So the other idea was that maybe DNA damage in these repair mutants would lead to some physiological state that would then favor additional signaling through any of those, those proteins. So we've done one experiment. We're irradiating worms and trying to see whether, if we induce DNA damage, and that seems negative, but we haven't tried other, you know, maybe more relevant, you know, missense inducing changes. So that's still kind of a puzzle. So, and it's not, the suppressors are, this goes back to Brenda, one of my really interesting inhibitors of the suppressors of the SWI genes, the one that we didn't test for amber suppression turned out to be an amber suppressor, as Brenda figured out when she joined the lab the first month.
First one I chose to study.
The first time I chose to study, right? So that was so that, but we so I've kind of, it turns out that the Cy15 mutation we started with is ochre. There's been never, you know, there's amber suppressor mutations that came out of genetic screens in C. elegans, but no ochre suppressors. So I thought, well, you know, maybe we have ochre suppressors, but they're not allele, they're not, you know, allele specific. So that's okay. That was the first thing I thought of. It's like, okay, you know, I don't want to get yelled at by Brenda again.
Oh, no, that it still hurts, but okay. So Priyati Sarwar is asking, there's a couple of related questions. One of your initial points of using the hypothetical G to P pipeline was to understand differentially expressed protein expression. What are your thoughts on how differential expression translates to importance of function, given there's examples of low expressed or low changing proteins that can have a large impact on function, and how to deal with that group of proteins that get missed from proteomics analyses?
So I think, all right, a couple of answers to that. So one thing is, I was always struck about how good microarrays were, okay, in terms of predicting biology. And, you know, as a technique, they're not so great, right? But I think what happened is that if you get a signal on a microarray, it meant there's a pretty big change, right? You know, that's like the twofold effect in a microarray, like, you know, we kind of make, you know, be nervous about it. And then these people you'd say, that's not going to work. That's ridiculous. And they come back and get some really nice biology out of it. And it turned out it's probably because it was so buffered that you're actually picking up things that are important. So, okay, from the omics approaches, I think what nobody's done is go down an abundance class and ask, okay, of all these hits, which ones matter, right? So for example, if you have 6,000 genes expressed in a cell, right, then if you go to the lower expressed ones, do they have function, right? So we really need a kind of a more systematic approach at the lower range to know whether we should ignore them and that their expression in that cell is noise or they're really important. You know, I don't know. I really do keep an open mind, right? And again, my initial criticism of the 10x genomic fad was, well, they're only getting 3,000 or 4,000 genes per cell, and we know there's 6,000. But then I thought, well, maybe it's like the microarrays, and then actually it's a good thing to focus only on the top 4,000, right?
And the more data you have, you can start doing comparative analyses and seeing, you know, co-expression profiles and things like that, which provide more information.
Yeah, and I don't know in yeast whether, you know, is there, you know, correlation the low expressed genes in a particular case in a phenotype, right? That would be the place where we could know that, right? No, I think one of the things, so in yeast, the one yeast experiment we've done in the last decade was looking at some RNA polymerase mutants and did an RNA-seq experiment with a sugar shift, so galactose to something else. And, you know, there's a third of the genome is changing when you change sugar, carbon source in yeast. It's not just the gal genes, it's lots, right? And I don't know if, you know, do those matter, right? I was hoping the Toronto group would, you know, crank through the genetic interaction studies, lots of conditions and really answer, you know, that question. That
we did a bit of that, but it's, you know, almost impossible to do large scale in, you know, hundreds of conditions. It's just, unless you have another type of assay.
Yeah, it's another type of assay.
We should ask, we can talk about it later.
Yeah, no, I'm saying I'm just saying that these are things like, to get that, if you propose that experiment, you know, it's not going to get funded, right? So this is a problem of, we're not systematic. But that would answer that question. I don't know. I think, you know, I'd love to say, you know, take something and just go to exhaustion and really try to understand where can we safely ignore it, right?
Yeah.
I mean, this comes back to evolutionary questions, which is, or thinking, which is, we know evolution is much more sensitive than we are in the lab, right? The 1% difference in phenotype we're not going to study, and in evolution, 100 generations takes care of that, right? So that's the problem with biology. So we have to be clever. We have to think about it, and we have to think about the organisms. Maybe also we can do comparative biology and genetics, because then each organism is emphasizing different types of genes. So a gene that is, essentially redundant or useless in C. elegans is really important in these other nematodes.
Yeah. Right. So just to follow up on that, do you view things such as stochasticity in gene expression, like rod selection in the eye, to be a potential barrier in genotype to phenotype predictions?
No, I think if you study those genes, you'll figure out where they're expressed and what the function is. And, really, it just takes more work, right? I mean, I think the stochasticity, especially it's programmed, is, you can solve, right?
And then just sort of getting back to the, you just mentioned sort of the evolutionary information that we can get because we're getting more and more genome sequences. Max Hyman says, most of us have more experience using mutant screens than cross-species comparisons to understand genotype-phenotype relationships. Should we be doing more cross-species comparisons? Do you have thoughts on tools, approaches that would help make this easier?
Okay, right now, telomere to telomere assemblies are actually, pipelined in with this Darwin tree of life. So I think that's good, and you want more of that. I think for animal genetics, we need a lot more genomes to get to the point where bacteria genomics got interesting. People said when there was several hundred genomes, but it was a lot easier because of gene clusters and to know which sets of genes would work. So you could look at genome content and get an idea. This kind of comes back to Jeremy's question about the myosin and actin, right? Like there are going to be changes. We don't actually know enough to know what's going to be regulatory and important. And I think that doing equivalent genetics in other organisms, is doable. We have one of these Steiner-Nema species, unfortunately doesn't jump, but it's hermaphroditic, and we can do nice CRISPR in that. And Meng-Yi Cao is a former postdoc in the lab's got carpocapsy CRISPR working nicely. But you have to, dig in. So I think there's, I think very focal experiments you could do cross species. So you have them, you can do the same experiment and it's, in real time and get an answer. It's very productive and interesting. If you start getting into like extensive screens and reconstructing the entire infrastructure, then it gets to be a lot of work, right?
Yeah, you might be able to think though about, so you were describing in one of your vignettes, the bypassing essential gene, I think it was the EGF receptor, right?
There was, I think the comparative biology, one case where we're looking at the effect of EGF on inducing vulval cells. And Wendy Katz said, well, there's a bias, like the posterior cells are responding less than the anterior cells. And it was kind of like, I don't know, 10% effect. And I convinced her to just ignore that for the time being, and that we could get back to that, to focus on the big question. So we did that, figured out the main thing. Meanwhile, Rolf Sommer, who was working on Proteomics and other species, had found that there was actually, we expected position-specific response to these inducing signals. And it was really striking. There was like even one species, Mesorhabditis, where the vulval precursor cells crawl to the tail, and then the only way the patterning could really happen, it wasn't independent of the anchor cell, had to be Hox gene regulation of their properties. So by looking at these other species, you go, oh, wow, that's really important. And then go back to C. elegans, and you could do the experiments, right, where there's incomplete penetrance. You have to do, 5 times as much work and get a clean answer. So I think that was really interesting, right? Yeah. Is you're going to, most of the time you go back to your favorite species and you can see hints of it, but a lot of times you wouldn't study it because it's too subtle, right?
Yeah. Exactly.
Right. We have to go for big effects, and that's the bias. And it comes back to the question about the gene expression, right? We need better ways to do assays so we can go for the smaller effects, right?
Yeah.
And,
Okay, I think we're gonna have to draw to a close pretty soon. We have a cool question from Anne-Marie Davison, who did lethal screens in the 80s and said optimistically in her thesis that we should soon figure out how all the genes build a worm. And how far away do you think we are from fulfilling Brenner's dream?
I think even though technology is accelerating, we have a long way to do it. I think we got to start, if you start thinking about the process and do you have a sufficient model? Most of the case, we don't, right? And the systematic experiments, we know how to do the experiments, but we don't do them. And they're not rewarded, right? Enough. So that's why I like, the micropublication idea, biology idea I like is good, because then you could do lots of, small experiments and get that information out there, and it kind of reward it. You can publish 1,000 papers, or one paper with a big table, right? But lots of information, right? Because I don't think we've actually, there's a long other conversation I'd love to have is, I think, as a biomedical research is actually underpowered, and we never really estimate what we need to do to solve the problem. And I think I would like us to start to figure out what we need to solve the problem. It's like building a bridge. We're building a bridge across a river, and we don't even know how far the river is or what the soil looks like underneath, right? Maybe we're too scared to know. And we have to just say, oh yeah, we can do it. We'll start building. It'll get there. Okay.
Yeah, thanks very much, Paul. It was really enjoyable, and thanks to everyone for attending.